This content is brought to you by Dr. Red's Virtual Labs. Sign up for free and start learning by doing.
Predicting reaction rates is crucial in chemical and biochemical engineering. Today, we'll explore best practices for identifying reaction kinetics parameters from experimental data. We'll use Dr. Red's Virtual Labs for data collection and Excel for data analysis. There are several methods for estimating parameters of kinetic models from experimental data. For many students, the differential method is often the first choice. While powerful, it has specific drawbacks you should be aware of. Let's briefly review the fundamentals of the differential method. As always, we begin by writing down the rate equation, also known as the rate law. Since we're unsure of the reaction order, alpha, and the rate constant, k, these parameters must be estimated from experimental data. Next, we turn to our virtual lab to set up an isothermal batch reactor and conduct a quick experiment. Because we used a batch reactor, the rate of change in the molar amount of reactant A is the product of the reaction rate and the reactor's volume. If we assume constant density, dividing by the volume yields the derivative of concentration. When we plug the rate equation into the mass balance, we get a differential equation with concentration as the independent variable. Theoretically, this equation could be integrated to find the concentration of A at any time, except that we don't know the values of the parameters. The differential method offers a solution: bypass integration. Instead, we manipulate both the equation and the data to simplify parameter estimation. The equation stays in its differential form, and we apply logarithms to both sides. If the term involving the logarithm on the left appears as a function of the logarithm of the concentration, a straight line results. The slope of this line corresponds to the reaction order, alpha, and the y-intercept gives us the logarithm of the rate constant, Ka. Let's proceed with Excel for our calculations. Taking the logarithm of each measured concentration is straightforward. But what about the time derivative? For simplicity, we approximate this by taking the difference in concentration between adjacent data points and dividing by the time gap. Whether you calculate this forwards or backwards in time, note that you'll end up with one fewer data point than you started with. Next, log transform the derived values. Now, we're set to plot the transformed experimental data. To estimate the parameters, add a linear trend line for slope and intercept values. For more accuracy, consider using Excel's slope and intercept functions directly in the cells. The reaction order is equal to the slope, and the rate constant can be found by taking the exponential of the intercept. Okay, that seemed easy enough. But what are the pitfalls in using the differential method? First, let's talk about the approximation for the time derivative of concentration. When your data has noise, using two data points to calculate the derivative can and will magnify those errors. To avoid this pitfall, you need to curate your data by removing outliers. This introduces a notable challenge. Initially, the measurement errors are randomly distributed. However, by selectively removing data points that exhibit a positive deviation from the true value, we inadvertently introduce a systematic bias into the error distribution. Consequently, the estimated value for the rate constant is likely to be an underestimate of its true value. The parameters we obtain are optimal for the transformed data, but not necessarily for the original dataset. The best way to ascertain this is to validate the model predictions against the original data. Utilize the rate constant and reaction order to simulate the batch reactor. Occasionally, suboptimal results with the differential method can be attributed to insufficient sampling frequency. The reaction rate generally diminishes as the reaction progresses. Therefore, consider increasing the number of samples taken early in the reaction when the rate is high. The final pitfall resides in the linearized mass balance equation itself. The fit to the data is unaffected by the value of the stoichiometric coefficient of the reactant. If you incorrectly utilize a stoichiometric coefficient of -1 instead of -2, you'll still obtain a seemingly satisfactory result. The outcome? Your rate constant will be inflated by a factor of 2, posing a significant risk in reactor design. Always write the rate equation carefully and substitute it into the mass balance to ensure your equation matches the reaction system under investigation. For optimal results, carefully plan your experiments. To span a sufficiently broad concentration range while maintaining a high sampling frequency, it's often best to conduct separate experiments under varying initial conditions. Curate each dataset individually and then combine them. In this example, the estimated parameters closely approximate the true values, despite the noisy data from the virtual lab experiments. Keep in mind, Dr. Red's virtual labs is not confined to batch reactors alone. A variety of single and tandem reactors, as well as CSTR cascades, are at your disposal for experimentation.